Okay, so I'll again thank the organizing committee for the second invitation to speak.
Sir?
All right, we'll do it that way. Okay, so
Are you okay with that bar on the top that's usually not there?
how do we get rid of that? Let's see. Okay, come on. How do we get rid of that?
Let's get back into the, when you got to this point here, what commands did you use? Icon to the right.
No.
Okay. So then try it again. Interesting. Okay. I'm going to have to troubleshoot that later and talk to your supervisor about
Okay,
that.
that's fine. That's fine. Okay, so let's start with a little bit of historical perspective. So the androgens actually were central to the, if you will, invention of the topic of endocrinology. And in 1849, Professor Berthold did some really classic, really the classic experiments, and in which he transplanted testes in roosters to show that the testes were releasing a humoral substance that was controlling the anatomy and behavior of the rooster, making the rooster act like a rooster, making the rooster look like a rooster. When he castrated the rooster, all that went away or didn't appear. And when he transplanted testes from another rooster, it reappeared. Okay, the behavior and the anatomy reappeared. So that was sort of the historical anchor to the topic of endocrinology, the subject, and showed that the testes were likely producing a humoral substance, a chemical substance that was affecting the structure and behavior of the rooster. Okay, so sometime later, about 40 years later, a scientist named Charles Edward Brown-Séquard, excuse my French, did some real classic studies where he actually, and he was older when he did this. I believe he was around 70, maybe older, and he was studying humoral effects of testicular extracts, and he actually was his own guinea pig. So he actually made these extracts from both dog and guinea pig, and he actually drank some of his extracts himself and reported that he felt much better, felt more vibrant, more energetic, just overall felt better. And this was sort of then termed the Brown-Séquard elixir. Okay, and so the idea was, testosterone, presumably, right? This was later assumed to be the effects of testosterone that was being extracted from the dog and guinea pig testes. It turns out much later that it probably wasn't the case because he was doing aqueous extracts of the testes, and so it probably wasn't really even testosterone he was ingesting, but it makes for an interesting story. Okay, so I guess we're doing it this way. So we talked about this yesterday, the established genomic effects of the androgens and the fact that not only do they affect differentiation and development of the reproductive tract, but have a number and effects on sexual behavior, but also have a number of anatomical effects throughout the body in terms of muscle mass and fat metabolism, bone growth and strength, and erythropoiesis. And all these effects can be addressed or are treated clinically with injection of testosterone or testosterone replacement in men. So, and to focus specifically on the cardiovascular effects of the androgens, of course, there's a very well-established dogmatic view that they exert as we talked about yesterday, deleterious effects on the cardiovascular system. The fact that there's a marked sexual dimorphism in human cardiovascular disease, it seems to occur more frequently in men than women. That it appears to exacerbate cardiovascular disease in men and contribute to things like atherogenesis in the vasculature, and clearly exacerbates hypertension in animal models. Now here's an interesting study, one of actually several that appeared in the 1940s, use of testosterone, propionate, and estrogenic substance in treatment of essential hypertension, angina, and peripheral vascular disease. Okay, this appeared in Journal of Clinical Endocrinology in 1942. okay, and it actually reported that testosterone injections improved the symptoms of cardiovascular disease in men, okay, and if you look at just sort of the summary of those studies, they all did very similar approaches, but basically intramuscular injections of testosterone were used. And we could say they are acute injections, but they probably were injections that were given and then over several days you could see improvement in the symptoms.
Did the estrogenic compounds not work in that paper?
They didn't work as well. And with those injections, they reported a successful treatment of angina, hypertension. So the angina, the pain from angina was reduced, hypertension was reduced, and peripheral vascular disease was improved. So circulation to the legs was improved. Okay, so we saw again and reduced systolic and diastolic blood pressure, and these effects were ascribed to direct vasodilatory effects on the coronary and systemic vasculature. Now they had no direct measurements in those days. They didn't have Doppler flow measurements or anything, or ultrasound or anything in which they could quantify that directly, but clearly from the alleviation of the symptoms suggested that there was a direct vasodilatory effect. So back to our studies now that we did years ago, looking at the Sprague-Dawley rats, males and females, and the testicular feminized rat. That's the rat that has the defective androgen receptor, so that it's the genomic effects, at least, of the androgens are not present. And I showed you data from these studies yesterday showing that androgens produce a rapid vasorelaxation or vasodilation of the isolated vasculature, that it occurs in both males and females, and is independent of the endothelium. It's an effect that appears on the vascular smooth muscle directly. It's a non-genomic effect that is independent of the cytosolic androgen receptor, okay, and because it occurs in the TFM rat as well as normal rats, both male and female. The effects are estrogen independent, and remember that androgens can be aromatized to estrogen in the periphery, and so theoretically it's possible that if you give particularly testosterone itself, that can be aromatized to estrogen, and that the effect could be estrogen, not androgen. Okay. Now that effect can be controlled for a number of different ways. One is to use different metabolites. For example, if you use dihydrotestosterone, that cannot be aromatized to estrogen. So if you see the effect with DHT, then you can be sure that it's not conversion to estrogen. And as I showed some data yesterday, these vasodilatory effects are structurally specific. Okay, so here are some of those data again, showing the filled squares, the nice dose-response relationship on the vasodilatory effect of testosterone, and in comparison the hemisuccinate ester, and that if we reduce the permeability of the ester across the cell membrane by conjugating to bovine serum albumin, it actually enhances the vasodilatory efficacy of the succinate compound. And here's some data I kind of blew by yesterday, showing that vasodilatory effect is present both as well in female and TFM rat aorta. Okay, and so this is a little bit different approach than that previous slide. In these experiments, we had a concentration response curve to phenylephrine, which is a vasoconstrictor. Okay, so it's going to cause the aorta to vasoconstrict. But what we did was we did a paired experiment in which we either added or not testosterone to the bath before we did the concentration response to phenylephrine. And you can see that in the presence of testosterone, the constrictor response is buffered by a good 50%. So this is just a single dose of testosterone that's added to the bath, single concentration, and so it's buffering or modulating the constrictor response, implying that it's a vasodilatory effect that is doing that. Okay, we see a very similar response both in the female rat aorta and in the TFM rat aorta. So very similar effect of testosterone to buffer or reduce, inhibit, if you will, the constrictor responses to phenylephrine. So more evidence that that the effect is non-genomic, that it occurs in females as well as males, and in that androgen-deficient TFM rat. And again, here's that figure showing the metabolic pathways that can result from testosterone that's secreted into the bloodstream. Again, it can be aromatized to estrogen. It can be converted to 5-alpha or 5-beta dihydrotestosterone. Okay, and I didn't make this clear yesterday perhaps, but the 5-alpha reductase enzyme is present in many of, in fact, I guess all of the reproductive tissues, and is the main mechanism by which testosterone exerts its reproductive effects. It's also present in other places such as the hair follicles, contributes to baldness, pattern baldness in the male. Let's see if we think of where else. Oh, I believe it's in the brain too, right? Is that right? Yeah. Okay, so it is present in the brain as well. Now the 5-beta reductase is that stereoisomer of this enzyme which produces 5-beta DHT. And you recall I mentioned yesterday that 5-beta DHT is genomically inactive. It's got zero activity as a genomic hormone, but we've been able to show that it's very active as a vasodilator. Okay, and so and the importance of this may be actually one important role may be in hypertension, because the levels of that hormone that enzyme are reduced in hypertensive individuals, and that enzyme and the 5-beta DHT compound are produced in the placenta. So we'll talk a little bit more about that in a moment. Okay, and then the excretory metabolic or breakdown metabolic pathways for testosterone, the dehydrogenase that produces the lipophilic 17-keto steroids, and then the more polar conjugates that are excreted in the urine by this pathway. So there's a number of different metabolic pathways that could contribute to actions of testosterone, including conversion to estrogen or the dihydro compounds. Keep wanting to do that. Okay, and again, here's that figure I showed yesterday with the difference in efficacy and potency comparing testosterone to the excretory metabolite androsterone here. The 5-alpha DHT, which is typically actually more potent as a genomic reproductive hormone than testosterone, but you can see here that 5-alpha DHT is actually less potent and efficacious as a dilator than testosterone, and that long-acting ester testosterone enanthate barely produces any vasodilation at all. So even though it's a really good long-acting androgen in vivo as a replacement hormone for androgenic effects, it's a very poor vasodilator. And we hypothesized again that the reason was that had to do with permeability to the cell membrane, that those esters such as enanthate, which are highly lipophilic, have a lot lower vasodilatory activity. Even though they're good as androgens, they're very poor as dilators. And you can compare them to other compounds such as testosterone, which are relatively less lipophilic and have a much better efficacy as a vasodilator. And again, the metabolites such as androsterone, which have zero genomic activity but fairly good activity as a vasodilator. So a really different structure-function relationship for these compounds. And we think, at least in part, that that has to do with the lipid solubility of the compound and its ability to work at the membrane as opposed to penetrating the membrane and getting into the smooth muscle cell. And also, just to reiterate that the data from the human umbilical artery, again, we've got different groups of androgens and progestins here. The progestins are actually pretty poor as dilators in general, but here you can see the dose-response relaxation effect of 5-alpha DHT. Again, just like in our studies, not as good a dilator. Here's testosterone itself, so a much better dilator, and about the same difference as we reported in the aorta, okay, comparing 5-alpha DHT to testosterone. And then much better, noticeably better, is 5-beta DHT, so it's markedly better than testosterone itself, okay, as a dilator. And that's, we think, reflecting its possible role as a vasodilator in placental circulation. Okay. Now, the data I didn't show you yesterday, here's a concentration response relationship that we did in the mesenteric microvasculature, and so you can see the concentrations here are much lower than they were in the aortic studies, so 0.1 to 100 nanomolar. And in the mesenteric microvasculature, pre-constricting the vessels with phenylephrine and then applying testosterone produced a nice dose-dependent, concentration-dependent relaxation response. The EC50 of this is somewhere around 3 nanomolar, and so that's well within the physiological range.
When you do those studies, is the vasculature like in the living animal not?
No, these are isolated
Or
vessels.
taking-
Yeah, these are isolated vessels. Right. Yeah, they're just in vitro in a tissue bath. Okay. Yes.
If I look on the curve for dose response curve for testosterone,
Yes.
it goes 1, 2, 3, 4 orders of magnitude and still creeping up.
Yeah. So,
So it doesn't look like a classical dose response curve where one would expect more or less full range of action within 2 orders of magnitude.
well, it's okay. It is 2 orders of magnitude: 1, 10, 100. 1 to 10.
Well, you start at 0.1
Yes.
and it goes is straight going up to 10,000.
No, to 10, or sorry, to 100, and then it peaks out. It peaks out at 100. Really, I think the highest we went up to was actually it may have been 100. I guess it was 100 or 500 nanomolar, but most of the dilator effect is down below at the lower range. Now, the important thing here is to note that when you do these studies in vitro, you have to impart some tone to the vessels because they're isolated. Okay, so in order to see a vasodilator response, they have to have some tone. Okay, and so you pre-constrict the vessels with some sort of vasoconstrictor. In this case, we used phenylephrine, which is an alpha adrenergic agonist, or you could use. We also did a comparison with high concentration potassium. Okay, so you can see that in the presence of phenylephrine, we get a nice dose response relaxation, vasodilation, but in the presence of potassium, high external potassium, 80 millimolar, that completely retards the relaxation response. The reason being, this is sort of evidence, functional evidence that potassium efflux from the smooth muscle is involved in the response, because when you have a high concentration, so normally the external concentration is about 5 millimolar for potassium, and when we jack it up to 80 to produce the pre-constriction, that almost completely eliminates the relaxation response because the efflux of potassium has been blocked by this much higher concentration. Okay, and we can show the same effect with a selective potassium channel inhibitor. So here's the patch clamp data that I didn't show yesterday, and so it's showing that again, this is in isolated mesenteric smooth muscle cells from the rat, and you can see under control conditions in the absence of testosterone that there's little current flow out of these voltage-dependent potassium channels, almost none, either under control conditions or in the presence of iberiotoxin. Iberiotoxin is a scorpion venom that blocks actually the calcium-dependent potassium channel. Okay, and so it's having little effect on its own, but those channels aren't very active under resting conditions anyway. And you can see that in the presence of testosterone, we get a nice run-up of the current flow, showing that those channels are opening more and more, or that they are open in the presence of testosterone. As we increase the membrane potential, there's more current flow out of the channels, as one would expect if they were open by testosterone. And in the presence of iberiotoxin, there's no inhibitory effect there. Okay, so we're getting a nice current, a nice membrane potential related flow, which is what you expect for a voltage-dependent channel. Now, when we pre-treat instead with 4-aminopyridine, which is a relatively selective inhibitor for the KV channel, you see that very dramatically squelches the current flow out of those channels. Okay, so again, at least some pharmacologic evidence with that patch clamp that the KV channel is involved.
Excuse me.
Yes.
Oh, sorry. So this is membrane potential, or is it a relative change in the membrane potential?
Yeah, relative change. Relative
Sure.
change. Right. Right. Okay. So, and I think I showed you this figure yesterday that's showing that we believe that part of the signal transduction mechanism here involves androgen's ability to activate nitric oxide production in the smooth muscle. And you can see under control conditions here some activity of nitric oxide in the mesenteric smooth muscle cells. And when we add testosterone, they really light up. So this is a nitric oxide specific fluorescence, DAF2DA. And so you can see with a relatively low 100 nanomolar concentration of testosterone that it really lights up the cells. So it's really turning on nitric oxide. Now the question might be what form of NOS is involved, or actually even before then, sort of a control slide to address the testosterone issue. Here's the cell under control conditions. Here it is pre-treated with ICI, which is an estrogen receptor antagonist, okay, a non-specific one that blocks both the ER alpha and ER beta receptors. And it has little or no effect by itself on nitric oxide fluorescence. And in the presence of testosterone with the ICI, we still get a nice increase in NO fluorescence, suggesting that the effect of testosterone is not mediated by its conversion to estrogen. If it were, then we would expect to not see the fluorescence here, okay, in the presence of ICI, and we clearly do. Now, this series, a time-dependent series of experiments shows that we believe that that source of NO is not from, well, of course, these are in isolated vascular smooth muscle cells, so there is no endothelium present. So it has to be another form of NOS, and we believe that it's neuronal NOS, which is constitutively expressed in smooth muscle cells. Okay, and so here are some cells under control conditions with the addition of LNPA, which is a relatively specific inhibitor for NNOS. You can see that over time, 10 minutes 10 minute pre-control, and then 0 minutes in the presence of LNPA and testosterone, 10 minutes, 15 minutes, there's no effective testosterone to light up these cells, okay? In fact, you can see some inhibition with just the presence of LNPA, so the neuronal NOS is evidently active somewhat under control conditions. And as a positive control, we then added sodium nitroprusside, which will produce nitric oxide in those cells, be hydrolyzed to nitric oxide in those cells, and you can see that they light up, showing that they are still able to produce nitric oxide, or respond to nitric oxide fluorescence. So probably probably NNOS is the compound is the enzyme that's involved. And again, I showed this model yesterday, which was really an earlier model, working model that we conceived of, where testosterone might either be acting directly on the KB channel, and this actually was derived from earlier studies of other steroids showing membrane effects, in which the steroids, and I think actually some neurosteroids, where they can actually, I believe, influence some of the channels in neurons directly. And so we thought that might be what was going on here. But then the more recent data we collected with the NNOS studies showed that probably there was most likely a membrane receptor involved that was activating NNOS, producing NO and cyclic GMP to activate our KB channel. So this was really an earlier working model. Now, an alternate model that's been proposed for these effects of the androgens has actually been inhibition of voltage-operated calcium channels in the smooth muscle. Okay, so that's another prominent channel in vascular smooth muscle, is the voltage-operated calcium channel. And so, so it's been proposed, and there is evidence, experimental evidence, showing that that androgen-induced vasodilation may involve blockade of extracellular calcium entry into smooth muscle cells, which would itself produce relaxation or vasodilation of those cells. Okay, and that it involves the L-type voltage-operated calcium channels, and perhaps also the non-L-type receptor-operated calcium channels. Okay, there's no evidence that androgens block intracellular calcium release, And a similar mechanism that is affecting the VOCC has been proposed or actually shown for estrogen-induced vasodilation, okay, that estrogen will actually inhibit the VOCC and cause relaxation that way. Now, a lot of this we think is probably concentration dependent, and so we proposed a similar model, as you saw in the last one, a similar early model where testosterone might be, again, acting directly on the VOCC as was proposed for estrogen to cause relaxation. So if you inhibit calcium entry, you're going to automatically cause the smooth muscle cell to relax and cause vasodilation. Now, really, so there's sort of a controversy that remains about the relative roles of potassium versus calcium channels in the vasodilator effect of the androgens. Okay, and so the issues really are first one of concentration, micromolar versus nanomolar concentrations. Okay, and so it seems as though the calcium effects occur at higher concentrations because a lot of the K-channel work that we've shown really is present at lower concentrations. And so it may be a concentration effect. Okay, it may be heterogeneity in the vascular preparation, that is whether we're using large conduit arteries which are less sensitive or smaller resistance arteries which are more sensitive to the effects of the androgens. Could be heterogeneity in the species that are studied, rat versus dog versus rabbit versus human. Also, another effect that's not often been well controlled in these studies is actually the interaction that occurs, the electrical interaction that occurs between the potassium and calcium channels. Okay, so that if I cause, because I can't get too far from the microphone. So if I activate the potassium channels, it's going to hyperpolarize or make the membrane potential more negative in those smooth muscle cells. If I do that, that is going to automatically close or reduce the activity of the voltage operated calcium channels. So you could see, if you ignored or weren't aware of the potassium effect, the potassium channel effect, the hyperpolarizing effect, you would see that or measure that pharmacologically as a reduction in voltage operated calcium channel function. And so it could be that the potassium effect is not well controlled for, and that is why some studies, that in concentration, why some studies report that the VOCC are mediating the effect. Okay. Now, another effect we noticed is that also that in some of our studies we noticed that if we in these isolated vessels, you have to sort of warm them up and get them stabilized. And so the way we typically do that is that they're contracted a couple times, vasoconstricted a couple times to kind of stabilize them and get them happy and stable. And when we did that with potassium, just adding high concentrations of potassium to the bath, it actually subsequently reduced the responsiveness to testosterone. We never could understand why this happened, but it was reproducible. If we pre-constricted several times with potassium, the testosterone, and then did a testosterone curve, they wouldn't respond very well. Okay. If we pre-constricted with another chemical agonist or hormonal agonist, such as phenylephrine or angiotensin, that was not a problem. That refractoriness never occurred. And so we never did get to the bottom of why that was, but clearly it was having an effect on the response pathway. So that could be an artifactual effect that's perhaps allowing the calcium channel effect to occur. Okay, so is there resolution possible for this difference? And so my answer is yes. I think there are. I think it is resolvable, and that both methods and physiology matter. Okay, so comparing the conduit versus resistance arterioles is one issue. The fact that the conduit vessels are always less sensitive than the smaller resistance arterioles, and of course, even just isolating a vessel, studying it in vitro tends to reduce its sensitivity, whether it's a large or a small vessel. Okay, the effect is a lot worse for the larger vessels because they're a lot thicker. Right, so the vascular wall is much thicker than a resistance arteriole, and it actually takes time for those steroids to penetrate the wall, get into the smooth muscle cells, and the large vessel walls are really a lot different in their structure. There's a lot more connective tissue, a lot more collagen, and the layers of smooth muscle are not as nicely organized as they are in a resistance vessel. So it really takes some time for the steroid to penetrate that and activate the cells. Okay? If you look at the time frame of those initial studies, it took us about 5 or 10 minutes to get a full response to the testosterone that's added to the bath. In the resistance vessels, a minute to 2 minutes to get a full response. So clearly the thickness and the heterogeneity of the tissue makes a difference in the responsiveness and the sensitivity. Okay. Okay, we talked about the interaction between potassium and calcium channels and the electrophysiology that's involved in that in the smooth muscle cell. The idea that potassium pre-constriction might suppress K-channel function somehow. And so what we propose is that all of these factors may contribute to this controversy in whether it's actually potassium or calcium channel that's involved. And also, this is probably the most important one, that there appears to be a biphasic effect of testosterone. At lower concentrations, clearly the potassium channels are involved. At higher concentrations, perhaps it's the calcium channels. Okay, so, and that plus the heterogeneity of the vessel size. So, let's see, and I showed you these studies yesterday, the translational studies on blood pressure. Again, the idea that we get a nice reduction in arterial blood pressure with IV infusions of testosterone in the conscious male rat. Okay, similar response in the TFM rat. So both Sprague-Dawson and TFM rats both respond with reductions in blood pressure. And I wanted to focus a little bit more on the comparing the 5-beta DHT response, which is in red here, and comparing that to testosterone. So just like we saw in vitro in the isolated vessels, 5-beta DHT is a better vasodilator in vivo in terms of reducing arterial blood pressure than is testosterone. And it's kind of interesting because the percent change here is almost identical to the difference that we saw in the umbilical artery in vitro. Okay, so the 5-beta is about 25% to 50% more efficacious as a vasodilator in vitro, and that's exactly what we see here in terms of the net reductions in blood pressure. Okay, now that may just be coincidental, but I think not. And actually, even if we look at the reductions in overall blood pressure, whether it's testosterone or 5-beta, this is the first study that's actually looked at mean arterial blood pressure with testosterone infusions given IV. There are several other studies that have done testosterone infusions into the coronary arteries. Okay, so anesthetized dogs, the heart was still in place, right? They just did infusions directly into the coronary artery. There's some other studies in pigs, anesthetized pigs, where they infused it IV, but just looked at, they never did measure blood pressure, unfortunately. They looked at regional blood flow, regional changes in blood flow. In both of those studies, they got almost the identical percent reduction, so about 25 to 30%, yeah, about 25% reduction in blood flow in those studies, okay, either to the heart or to the various vascular beds, which is almost identical to what we get, about 25 to 30% reduction in blood pressure. So that effect seems to be pretty reproducible from dog to pig to rat, and also I should mention that those studies have been done. Some studies have been actually done in humans where they infused testosterone in the coronary arteries and saw dilation. So it seems to be a pretty reproducible effect, and one that would be significant in terms of reducing blood pressure. Now this 5-beta is interesting because, again, it's a compound which shows no genomic activity as an androgen, but it's actually better as a vasodilator compared to testosterone. So we're currently pursuing this hypothesis that it might be, because 5-beta is produced in significant amounts by the placenta, that it might actually play a role in helping to maintain placental blood flow. Okay, and so to sort of get at that hypothesis, we need to actually do some studies where we measure, I think, changes in 5-beta production by the placenta, but we're using a model of preeclampsia, which is pregnancy-induced hypertension, in which the hallmark of that is hypertension and vasoconstriction and reduction of blood flow to the placenta and to the fetus, and so, which is a big issue because it's very deleterious to the survival of the fetus, right? So the blood flow directly affects the fetal development, and in preeclampsia, one of the things you see is significant retardation of fetal growth and development because of the impairment of blood flow. And so we're doing studies right now where we've established the preeclampsia model in our rats, and we're looking at reactivity, at least as a first test or first arm of the study, looking at potential changes in reactivity of the umbilical and uterine arteries to 5-beta-DHT to see if there might be a difference in reactivity of the compound as opposed to change in the production. It could be one or both of those issues that are involved. Let's see. So, okay. So this was an earlier model that we proposed a few years ago that just sort of compared effects of the androgens on both genomic and non-genomic actions of the androgens on vascular smooth muscle. So here we have sort of a schematic blood vessel, if you will. The genomic effects being mediated by the androgen receptor, and so you can see 5-alpha DHT, which is much more efficacious and potent than testosterone, but they both interact with the same androgen receptor in the cytosol, translocate to the nucleus to modulate transcription and translation of DNA, and which results in the androgenic responses that we see in the male animal. Okay. The non-genomic effects, here's the vascular smooth muscle cell membrane, and so again, showing potentially a closing of the calcium channels, which would cause relaxation. A very few studies, I think about 2, have shown actually that acute testosterone administration can actually cause vasoconstriction, so it's possible that it could act to open those channels. For the sake of completion, we showed that in this model, but I don't think it's very likely. I think it's probably by now an artifact. I mean, there are dozens and dozens of studies now showing that it clearly produces vasodilation. So I'm not sure what's going on there, but and then thirdly, here's our effects to increase cyclic G or cyclic AMP and open our potassium channels and cause potassium efflux to cause our non-genomic relaxation. And I showed you this slide yesterday, a little bit more recent, actually from just a couple years ago, a little more recent picture of the signal transduction mechanism, mainly focused on the vascular smooth muscle and the ability of testosterone to perhaps interact with a membrane androgen receptor on the smooth muscle and go through the PI3 and AKT pathway to activate NOS, nNOS, and form cyclic GMP, activating protein kinase G, which activates, in this case, this is in coronary vascular smooth muscle, showing an activation of that high conductivity, calcium-dependent potassium channel to cause the vasorelaxation. And again, at the same time, it's probably closing the voltage-operated calcium channels to cause relaxation. Okay, so I've hopefully convinced you that we do have a beneficial non-genomic vasodilatory effect here, that it's endothelium independent, working directly on the vascular smooth muscle. It's a structurally specific effect, okay, that involves modulation of ion channel function in the smooth muscle and appears to be mediated, at least in part, through the nitric oxide cyclic GMP pathway. And again, the idea of finishing up, looking at where this would go in the future, the idea that the development of selective androgen receptor modulators, the SARMs, which has really been a lot more recent than the formation of the SERMs, the estrogen receptor modulators. That's much more advanced, and of course they've been used clinically for years now. Actually, I think we're on the 3rd or 4th generation modifications of those SERM compounds, starting with tamoxifen and moving to raloxifene and some of the more recent ones. That I think it's sort of, that work is sort of stalled. They haven't been able to they haven't achieved as much success as we had hoped in terms of the selectivity of those compounds. But and interestingly, sort of a side note, we did some studies on this a long while back, but actually showed that tamoxifen, even though it's largely an estrogen receptor antagonist, does have some agonist activity, and actually in terms of activating the thromboxane pathway and activating vasoconstriction, it actually does that. Tamoxifen does a very similar thing. It boosts the vasoconstrictor effects in the vasculature. And so at least in the cardiovascular side, it's actually a bad thing because it's activating those thromboxane pathways. I'm not sure about raloxifene, I don't know that anybody's tested that one for for this mechanism or not, but tamoxifen clearly does that. So it's kind of interesting. So with the SARMs, a few of these have been synthesized, and the idea would be that we could make one with no genomic activity similar to 5-beta DHT, and that it might be used for therapy for hypertension or coronary artery disease to promote vasodilation systemically or even regionally to treat erectile dysfunction, or in the female, pregnant female, to treat preeclampsia or other vascular dysfunctions. Okay, and these would this would be good if we had these selective compounds without genomic activity, because we wouldn't have any of the androgenic side effects. All right, and we'd still be able to target the cardiovascular system to produce vasodilation and treat these vascular dysfunctions. Okay, but still, there's a role, I think, for the androgens working through the androgen receptor to treat and improve health in hypogonad aging men, because it's clear that androgens actually have an overall beneficial effect in these aging men. And as I talked a little bit about yesterday, I think the discrepancy here between are the androgens exacerbating cardiovascular disease or not, I don't think they are. I think what's happening is that it's the loss of, it's the effects, combined effects of age, and that's actually producing the problems in cardiovascular function, and the gradual fall in androgens with aging and even normal men. So that a lot of the cardiovascular disease you see in men that are 60 or 70 is probably not so much a history of having androgens on board as much as it is an issue of aging and the loss of androgens over time. Because when you replace androgens in an aging hypogonadal man, you improve the lipid profile, you lower the blood pressure, you produce an overall beneficial effect on their metabolic health. And so that's not consistent with the idea that androgens are deleterious. I think it's the aging process and the loss of androgens over time that's resulting in those apparently deleterious effects of the androgens. Okay, so specifically the collaborators that contributed to the androgen studies, Richard White and Mercedes Peresquia, who's my collaborator at the National University in Mexico, and postdocs and graduate students that have contributed to this work as well, and support from NIH. Okay, thank you.
So I'm wondering, what do you think about the role that some of these SARMs might have in altering the way testosterone would work in the central nervous system, or the interaction of conditions that would provoke hypertension from the brain?
Yeah, that's certainly possible. I mean, and again, I guess it could be, from all that evidence of other steroid effects in the brain, non-receptor, non-genomic steroid effects on nerve cells or even glial cells. Sure, it could be either. It could be genomic effects that are being that are affecting that, or non-genomic effects which are modulating blood pressure. I mean, it's entirely possible. Absolutely.
So did anyone ever give this alpha compound,
the 5-beta DHT to the brain?
And see if it did anything in the brain? That would be interesting.
Yeah, it would. It would. It'd be very interesting. Yeah. Yes.
I'm slightly intrigued by the slide where you showed that you have membrane androgen receptor. The reason why I'm asking is now I'm looking for the ways how progesterone acts in a non-genomic way.
Mm-hmm.
And most of the literature I went to, they suggest either it is through a receptor kinase receptor, or through some growth factor means it acts through the membrane. So now in your case you are telling that it's a membrane-bound androgen receptor. So how different it is from the conventional androgen receptor inside the cytoplasm? It's almost same, or after binding, whether it translocates to the cytoplasm and then brings about the sequence? So
the one membrane androgen receptor that's been identified is a G protein coupled receptor. Okay, so it doesn't translocate, but it produces a second messenger. But as I mentioned yesterday, that membrane, as far as we know, that receptor, as far as we know, is not mediating these dilator effects because it's causing an influx of calcium. which would produce vasoconstriction if it were present in the smooth muscle cell. So it's got to be a different receptor. We just don't know what it is yet.
Okay, and my second question
Yes.
is, again, you said that the some positive effects of the testosterone administration, especially decreasing blood pressure in rats, dogs, and pigs. But I'm just here alluding to the fact that the clinical trial with the normal men with the normal testosterone, they gave testosterone and then they found out that one person died because of myocardial infarction, and then they stopped. And they observed changes in lipoprotein profiles also. So with that in mind now, I don't know how you can extend your observation in the animals to the one in the human study. So is there any beneficial?
Yeah, I mean, that's actually, so there have been a few clinical trials where that has occurred, but there's also been a lot of data more recently, meta-analysis, that was what I was trying to remember yesterday, meta-analyses that have been done to show that there's no clear, measurable effect of the androgens to increase coronary artery problems or mortality. So if you look at the broad collection of data that's available, there are those studies, and I don't know whether it's the form of the androgen, the age of the patient, or what exactly is going on there. But I agree, there are a few studies that suggest that there is an increase in mortality, but overall, when you look at the meta-analyses, there's no clear evidence that it is, that it is increasing mortality. And in fact, it may be very dose dependent, because there's a very recent paper in the American Journal of Physiology that summarizes some of those recent meta-analyses, and they show quite clearly that it's very, very dose dependent. At the lower and moderate doses, there's significant beneficial effects, but no increase in mortality whatsoever. In fact, there was actually a slight reduction in mortality. Yeah. Okay. Yes?
I am an organic chemist, so I just...
Good. That's actually good. I'm glad.
So I just want to make a comment, okay, about testosterone and 5-alpha and 5-beta
Yes.
DHT. Okay?
Yes.
So testosterone is more or less flat.
Yes.
Okay. 5-alpha DHT is also more or less flat. Okay?
Yes.
But 5-beta DHT is not flat.
Correct.
So, okay. So I am not so surprised that the biological activity of these compounds, 5-alpha and 5-beta, is not the same.
Yes.
That's what you have seen. That's probably due to the configuration of the molecule.
Exactly.
It's really a huge difference.
Yeah. So that alpha. So it's right. The alpha ring, or the alpha.
The alpha substituent at the
Right.
fixed position.
Right.
This is flat 5-alpha. Okay.
Right.
But 5-beta is so.
Right. It's like
it's alcoholic acid.
Yeah, so it's bent, like almost.
It's bent,
yeah, it's
exactly.
very bent. So yeah, so you would expect molecularly
Yeah.
that it's going to have a different effect.
Yes.
Exactly.
Yes. So you can expect a different biological activity.
Right. Exactly.
You can
Exactly.
expect.
Is that true also of the 17-beta estradiol, 17-alpha estradiol? Because I know in the old literature, they use the alpha as a control for the non-estrogen receptor mediated action.
I think that general remark for the steroids, okay? As soon as you modify configuration of whatever carbon, 17, 5, 3, whatever, okay, then you change a lot of things on the molecule, okay? And the biological activity, I expect that the biological activity should be different, or the molecule doesn't have the same properties.
I see.
Because in space, it looks like really different. Okay?
So I think that the 17 alpha and beta forms,
it's less important. I
it's
think.
less important. I think there's less of a change in the.
Because it's on the side chain. Excuse me.
Right. Yeah. No, you're exactly right. So so it's in that change is in a different part of the molecule.
Right.
Not at the tail, not at the front end.
Exactly.
It's somewhere in the middle, and it's a side chain.
Yeah.
Whereas with the 5-alpha and 5-beta, it's the alpha ring, which is actually bent.
Yeah.
Yeah.
Whole world. But then you wonder, what else will it bind to if it doesn't bind to the androgen receptor? Are there any other receptors?
Oh yeah, sure,
that
and that's
bind to?
sure. That's entirely possible.
And that would be
Yeah.
interesting.
Yeah. Yeah.
You know.
absolutely.
Right.
All right, any other questions? All right, thank you very much.
So if that clock on there is correct, is it really 10:36?
It's
Yeah, it
11:36.
11:36. All right, we have
Okay.
a break.
Yeah.
We'll take a break
Okay.
and